Today we'll be looking at a Unit 4 FRQ practice. So this practice problem is really about cell signaling. It's also a little bit about like DNA and mutations, which we haven't covered yet, but the primary focus of this FRQ is on cell signaling. I picked this FRQ because I think it's extremely difficult, and if you can master this one, especially after me talking you through the problem, I think you'll be very successful on many FRQs. So the plan for right now is I'm going to have you pause the video in just a second and read the FRQ. I want you to circle keywords you see. I want you to write definitions for things you know. I want you to write question marks for things you don't know. I want you to read the entire problem. I want you to look at the diagram. I want you to read questions A, B, C, and D and kind of highlight things. So you're going to do that first. Then you're going to unpause the video and I'm going to read through the problem and make notes about things I notice. Then you'll pause the video again and answer the questions. Then you'll unpause and listen to me go over the answers with you. Okay? So make sure you follow the directions as you go, and then you'll really get a good look at how I would think about the question, and also think about how you would think through the question. So at this point, I want you to read the question and look at the answers, look at the questions and think about how you might answer them. So pause the video now. Pause the video. All right, now you're going to unpause the video. Hopefully, well, if it's paused, you won't be able to hear that. But after you read it, unpause the video. And now I'm going to talk you through my thoughts on this question. So when I read this question, okay, I notice that it's talking about the eyes of arthropods, okay? And they contain different types of cells, photoreceptor cells and cone cells. And you might not know what those types of cells are, but that doesn't matter for answering the question. Scientists study how cells interact together and how that affects the developing eyes, okay? And it says that normally these R7 precursor cells become photoreceptor cells. So these are kind of the before cells and these are the after cells, okay? But then scientists notice that there are two cells located in the developing eyes, okay? And they have different types of proteins. R8 cells, so these R8 cells right here, have a protein called BOS sticking out of them, okay? And our 7 cells right here, the precursor cells, have this tyrosine kinase receptor. So what am I noticing right here? I'm noticing a type of cell signaling, and I'm going to make myself a note of it. This is a type of cell signaling where the two cells are touching each other, okay? And this is called cell-to-cell or juxtacrine signaling. So this is a type of cell signaling I recognize, and I can even look at my big sheet to verify, yep, when the two cells are touching, that can happen. And actually, it talks about, one of the examples is neural development, and this is eye cell development. So when I read this, it looks like this is a tyrosine kinase receptor. And what do I know about tyrosine kinase receptors? Okay, I know that tyrosine kinase receptors can signal multiple pathways inside a cell, okay? Ooh, and this is interesting. This tyrosine kinase receptor activates a G protein called RAS inside the cell. So this RAS right here is a G protein. And you can kind of see it changing shape here, right? The BOS was just this protein ligand, and it's interesting because BOS is a protein ligand that is apparently attached to the R8 cell. So this ligand attaches to the cell surface receptor, the SEV-RTK, okay? And that activates this RAS protein. You can see here's its inactive form and here's its active form. And then that looks like that leads to different signaling pathways inside the cell, which we know because we know tyrosine kinases can activate multiple pathways. Okay, excellent. Now, how does it do that? Let's look inside the reading to see how it does that. Okay, the RTKs bind to the ligand, yep, to the BOS, and an intracellular portion of this RTK transfers a phosphate from ATP to other proteins. So it looks like SEV-RTK transfers a phosphate to RAS. And we know that when we transfer a phosphate, that's called phosphorylation. So it looks like it activates that protein. Okay. And then it looks like they did different experiments. So let's look at the experiments down here. In this first experiment, this is wild type. Wild type, by the way, means like the normal type found in nature without mutations. Okay. Oh, also another thing that I notice in here is they keep talking about differentiation. What is differentiation? Differentiation is becoming a specific cell type. And you can see that here. In this example, okay, when this ligand, this boss ligand attached to this R8 cell attaches to the cell surface receptor on this precursor cell, this isn't the final cell, then this is probably like some sort of stem type cell, okay? This activates ROS by phosphorylating it, so we're starting a phosphorylation cascade, activating multiple signaling pathways, so multiple transduction and response pathways. And then the response, the outcome, is this becomes a photoreceptor cell. So differentiation has occurred. This has become a specific cell type. Okay. In example number 2, it says there's a mutation in SEV. So if SEV is mutated, that typically means that SEV is not working. And you can actually see this in the picture. You can see here how the bottom of SEV has this kind of like straight cylindrical shape. And here how the bottom of SEV has bulged out. So that is due to the mutation. There's been a change. I'm going to write it over here. Sorry. A change in protein shape. So let's look at the consequence of that. Due to this mutation, it looks like RAS is unable to bind to this tyrosine kinase receptor. So if you can tell, it's in its inactive form. See, it's in its inactive form. And no signaling pathway occurs. So we have no signaling here because the signal is lost, right? This receptor has changed shape. It can't bind to RAS and activate it, no signaling. And in the absence of signaling, we become a cone cell. So it looks like if signaling occurs, we become a photoreceptor cell. If signaling in this pathway doesn't occur, we become a cone cell. Okay. And then let's look at this third example right here. This is a double mutant. So you have the SEV negative. That means the RTK protein is mutated. It's not working, right? But the second mutation is really interesting. The second mutation is an activated RAS. So what does that mean? Let's look in the reading. ROS-D is a form that is always active. So interestingly, it looks like this is always active. So this is in its active form, not its inactive form. So they're wondering what is happening. So like, is there going to be no signaling because this is mutated? Or is there going to be signaling because this is in its permanently active form? So that's something you have to decide. I think that's in one of the questions. Okay. And they actually say in the figure, we should always look at the title first. I should have done that. What is the fate? What is the outcome of these 3 cells in different environments? Wild type, when the receptor is mutated, when the receptor is mutated, but RAS is continuously on. Okay. So now what I'd like you to do is read through the 4 questions, write down your answers, what you think your answers are. Okay. And then, while you're doing that, you should pause the video. When you're done writing down the answers, you're going to unpause the video and I'll go over the answer key with you and what I would say. Okay, so at this point, pause the video. All right, now you should have unpaused the video and you should have written your answers down for this question. So we're going to go over the answers together. And I left you some space. Hopefully you used it back here to answer the questions. So let's look at A. I'm going to think through what I would say. And then actually, I just printed out the answer key, but I haven't looked at it yet. So we'll see how close I am to the actual answer key. Okay. A, describe the first interaction that must occur between the R7 precursor cell. So I'm going to put my finger on it. Oh, sorry, the R7 precursor cell. I put my finger on the wrong thing. And the R8 cell for the R7 cell to differentiate, meaning to change into a photoreceptor cell. Okay, so what's the first thing that needs to happen? This makes me think about my signal transduction pathway. You have to have reception, you have to have transduction, and then your response in this case is turning into a photoreceptor cell. So the first interaction has to be about reception. And the very first thing that happens is it looks like this ligand bound to this receptor. So I would say for this one, the protein ligand binds to the cell surface receptor, SEV-RTK. That would be what I would say. Let's see what they said in the answer key. Let's see. They said, okay, oh, phew. I should have looked at this answer key before I did this, but I wanted it to be authentic. The response indicates that the SEV-RTK on the R7 precursor cell binds with BOS on the R8 cell. So if you said that, give yourself 1 point. All right, let's look at part B. Describe how the defect in the single mutant SEV minus R7 precursor cell causes the cell to become a cone cell instead of a photoreceptor cell. So I think we kind of talked about this already. I would say something like, because the SEV minus the tyrosine kinase receptor is mutated, BOS can still bind to the tyrosine kinase receptor here, but because of the mutation on the internal part of the protein, ROS can no longer bind to the tyrosine kinase receptor. That means ROS can't be activated and you'll have no signaling. And it seems like no signaling results in a cone cell. Let's see what the answer key says. I probably said too much there. Okay. The answer key says that the response indicates that the RAS protein cannot bind to the mutated intercellular portion of SEV. Good, I said that. And become phosphorylated or activated. Ooh, I forgot the phosphorylation part. So RAS does not induce the intracellular signaling pathways. Ooh, that's a good keyword, intracellular, necessary for differentiation of the R7 cell to the photoreceptor cell. Like I said, for the R7 cell becoming a photoreceptor cell, but this keyword differentiation is important here. All right, let's look at part C on the back. The scientists predict that the presence of the activated RAS in the double mutant precursor cell will enable the cell to differentiate into an R7 photoreceptor cell. Based on figure 1, evaluate the likely accuracy of this prediction. So when they say evaluate, okay, they want you to say, is this prediction, do you agree with this prediction or not, and why? So do you agree or disagree with the prediction, and why? That's what evaluate means. Okay, so let me look at this. I would agree with this prediction. I would agree that this cell down here is going to turn into a photoreceptor cell. The reason I would agree with this prediction is because, yes, this receptor protein is mutated, so it cannot independently activate RAS, but RAS is activated on its own. And we know from this first picture that if RAS is activated on its own, it's going to trigger transduction and response signaling, okay? So I think it is going to trigger the signaling pathway because it doesn't need to be activated by the receptor, and you're going to get a photoreceptor cell. Okay, let's see what the answer key says. The answer key says that the response indicates that the prediction is most likely supported by the data. Ooh, I should have said supported by the data. Because the RAS protein is already activated, it does not need to become phosphorylated or activated by binding to the intracellular portion of SEV. So RAS can signal, and the R7 precursor cell should become an R7 photoreceptor cell. Okay, basically what I said. But again, the supporting by the data, that's a good point there. Okay, let's look at part D here. Part D. The SEV-RTK, so the tyrosine kinase receptor, is expressed. Okay, we haven't talked about this yet. Expressed means the protein is made in more cell types than just R7, and it appears to be important in determining the phenotypes of the cells. That means like what proteins are made in the cells, what the cell looks like. So like cell structure and function is the phenotype, right? Explain how one receptor can induce different phenotypes in different cell types. Well, first of all, the receptor here, okay, this receptor is responsible for activating some sort of intracellular protein, in this case, RAS. This receptor may be in a different protein, can activate a different second messenger or a different G protein, right? Or it can trigger a different phosphorylation cascade. We also know in these tyrosine kinase receptors that they can activate multiple signaling pathways. So maybe in other cells, this RTK, this tyrosine kinase receptor, binds to a ligand, maybe a similar ligand, but while the reception part is different, there's different phosphorylation cascade or cell second messenger signals that lead to different responses. So there's different transduction and response inside the cell. Let's see what they say. The response indicated that the proteins that interact with or are activated by the or phosphorylated by the SEV-RTK in different cell types may differ. Yes, so different proteins, so different phosphorylation cascades. Different ones could be activated in different cell types, or the proteins are activated later and the intracellular signaling pathway may differ. So basically what they're saying is it could activate different second messengers or different parts of reception, right, other than the G protein. So reception could be different, okay? Different proteins could be phosphorylated, or proteins that are involved in transduction, different proteins could be activated during transduction, which could lead to different responses. All right, so add up your points. See how many did you got? Did you get 1, 2, 3, or 4? This is a difficult question. Thank you for working through this one with me, and I hope this was helpful.